Hello everyone. One of the most popular requests for videos that I've had is for the carriage of oxygen in the body. Now, I've done one on the carriage of carbon dioxide, and this really goes kind of hand in hand with that one. It's a key stage 5 biology revision video that I'm doing here, all about hemoglobin and the oxygen dissociation curve, or more specifically, the S-shaped Oxyhemoglobin dissociation curve. So I'm going to also make reference to, in this, fetal hemoglobin, and we're going to look at an exam question as part of it. Now, before I talk about hemoglobin and the carriage of oxygen, I think it's important just to acknowledge the passage that air takes in the body, because for that oxygen to get to the hemoglobin, it's got to first go down the trachea, then the bronchi, then the bronchioles, then the alveoli, through into the plasma, through to the red blood cell, and then to the hemoglobin. So it's quite a passage that this oxygen molecule has to take. And then we get to the hemoglobin. So this is where this story really picks up. Now, hemoglobin is a quaternary protein. By quaternary protein, what I mean is that it's made of 4 polypeptide chains. And I'll highlight these on this particular diagram that I've got here. So you can see we've got a beta chain 1 here, a beta chain 2. We've got an alpha chain 1 and an alpha chain 2. So when you learn about protein structure, you've got a primary sequence, the sequence of amino acids, that develops into a secondary, which is an alpha helix or a beta pleated sheet. You've got your tertiary folding, and then there's this other level, this quaternary structure, when you have multiple polypeptide chains. And that's what we have here, these 4 polypeptide chains. Now, each chain contains a heme group. Now, it's spelled on here H-E-M-E. I've spelled it in the past H-A-E-M. All essentially the same thing, but what we've got is this heme group here, and the red dot represents, I think my green pen's just gone over it slightly, is an Fe2 plus ion, an iron ion, Fe being the symbol for iron. So that's really important that we've got this Fe2 plus ion there as part of the heme group. And I'll just make a note, as I said, that this is a quaternary protein. And by quaternary protein, I essentially mean it's got multiple polypeptide chains, and this one has 4. 1, 2, 3, 4. Now, the heme group has a high affinity, or attraction as we call it, for oxygen. Each one can actually carry 1 oxygen molecule. So that's something we just need to put into place. The right side of this diagram labelled B, so here, all of this is essentially just a structural formula for this, but I'm not necessarily going to refer to it. I've put it up there just, not just for a visual, but for reference. But in this video, I'm just going to talk about the actual diagram to the left. So as I said, this Fe2 plus ion has an affinity for 1 oxygen molecule. So each one has an affinity, and this word affinity is one we're going to use a lot in this video. It means attraction, and it's an affinity for 1 oxygen molecule. Now, oxyhemoglobin is formed by a reversible reaction in the lungs where you get oxygen plus hemoglobin. So oxygen plus hemoglobin forms oxyhemoglobin. Then when blood reaches the body tissues, it turns back to hemoglobin as the oxygen leaves or disassociates from the heme group. Now, a key idea that we need to address before we really talk about the oxygen dissociation curve is about the partial pressure of oxygen. So we're going to talk about the partial pressure of oxygen. Now, the partial pressure is often referred to as the oxygen tension. So if you see that in a book or on a website, that's what we're referring to. The oxygen tension is essentially the same thing. So we're going to talk about the partial pressure of oxygen. Now, the partial pressure of oxygen, and it's often written as P, a small p, O2. So the P here that I've done represents partial pressure. The PO2 of oxygen, it's a measure of the pressure that oxygen exerts in a mixture of gases. So we'll just make a note, because that's one question that often comes up as a definition. It's a measure of the pressure oxygen exerts in a mixture of gases. And it's used as an indirect measure of its concentration. And I'm using that word, if I wrote it, it would be in inverted commas, so just be careful. So it's a measure of the pressure that oxygen exerts in a mixture of gases, but it's indirectly, we use it as a measure of its concentration, if you like. Now, hemoglobin picks up oxygen in the lungs where there is a high PO2, high partial pressure, and it delivers the oxygen to respiring tissues where the PO2, the partial pressure of oxygen, is much lower, as the cells use up oxygen in respiration. So just to be clear, where there's a high partial pressure, where there's a high concentration, if you like, of oxygen, the hemoglobin wants to pick it up. And where the partial pressure of oxygen is low, so in the respiring tissues that ultimately need it, the hemoglobin is designed to release it for the oxygen to dissociate with that heme group. So let's look at what's called the oxygen dissociation graph. Now for this one, I'm going to shrink the screen a little bit here. In fact, we'll move this right up to the top. And we're going to work over on the right-hand side. Now, as the partial pressure increases, the saturation of hemoglobin increases. And one question you may get asked is to predict what an oxygen dissociation graph would therefore look like. Now, the answer to that is essentially this that I'm just about to draw. So I want you to imagine we've got a graph like this. We'll take most of the page. We're going to have the PO2, the partial pressure of oxygen, on the bottom. And the units, we'll put the units because that has a unit of kilopascals. Remember, it's a pressure, and pressure is measured in pascals. We're going to have the percentage saturation of hemoglobin as our y-axis. So we're going to put it here. Percentage sat for saturation of hemoglobin. There. Now what you'll get is a shaped curve that looks like this. Notice there's a very subtle S shape, and this is called the S-shaped Oxyhemoglobin dissociation curve. Now, one key thing to state is that the conformation of the hemoglobin molecule changes, so the shape changes as an oxygen molecule becomes associated with it, altering its ability to associate others. So let's start at the bottom. The heme group that attracts the first oxygen is in the centre of the hemoglobin molecule, and it makes it quite difficult to bind. And that is what accounts for the low saturation level at a low oxygen tension, or a low partial pressure of oxygen. So if we have the red circle as that's representing one molecule of oxygen binding at a low partial pressure of oxygen, let's just put some numbers to this. Let's say we've got 2, 4, 6, 8, let's go there. Let's say here we're looking at 20, put 40 here, 60 we'll have over here, 80 here, 100 at the top. Now clearly it's not to scale, but it's just to put some values onto it. So again, we said that the first oxygen is quite difficult to bind because it's in the centre. The heme group is in the centre of the hemoglobin molecule. So that's why we've got a low saturation of hemoglobin at a low partial pressure. Then, what we can do is put the second one in place. We imagine 2 oxygens were to bind, or the second oxygen rather were to bind. Now after the first oxygen molecule associates, conformational change makes it easier for the second and the third oxygen molecules to associate. So we can even put the third one up now. So we've had the second oxygen molecule bind, and now we've got the third one. But by that point, it's quite difficult now to associate a fourth oxygen molecule. And that's because we say the hemoglobin becomes, again, in an odd way, full. The curve starts to plateau below 100% saturation. So really, my blue line shouldn't necessarily have gone so high. It should have maybe plateaued about here. So just below 100. If we take 100 as the top of the graph here, we're plateauing just before. So let's just, for completeness sake, let's put our 4 oxygen molecules in place here. And as I said, it's difficult to associate a fourth oxygen molecule because the hemoglobin becomes full. The curve plateaus below 100. Now the oxygen tension in the lungs is sufficient to produce almost 100% saturation. The oxygen tension in respiring tissues is sufficiently low to cause it to dissociate readily from the oxyhemoglobin. So you've got to keep that idea in mind. So the first one binds rather difficult. So we'll just put a cross there. These two bind fairly easily. And then this fourth one we'll put a cross, just to remember it's a little bit more difficult to have that binding. And this is our classic S-shaped oxyhemoglobin dissociation curve. So in the lungs, where the partial pressure of oxygen is high, what we're saying is that the hemoglobin can saturate with oxygen. It's able to pick up a lot of the oxygen molecules. So for a high, if we just use this graph, for a high PO2, just picking a random value here, we can see that the oxygen or hemoglobin rather, saturation is high. We're able to associate with the hemoglobin quite easily. In the respiring tissues, where the PO2 is a lot lower, you can see that the saturation of hemoglobin is a lot lower. It's much harder to associate the oxygen. In fact, quite the reverse. It makes it easier, the body makes it easier to dissociate readily from the hemoglobin. So we break down that oxyhemoglobin, it gives up that oxygen far easier. Now just an extra overlapping layer on this, if you like. We're going to talk about fetal hemoglobin, because this often comes up in exams as a question. Now the question that often gets asked is, why would a curve for fetal hemoglobin shift to the left? So this one here, if we say this is for adult hemoglobin, what you get is something that looks a bit like this. For fetal hemoglobin, so I'll just label that in place. So that curve there represents fetal hemoglobin on this oxygen dissociation curve. Now the reason why, or the answer, is that to get oxygen from maternal to fetal blood, fetal hemoglobin needs a much stronger affinity for oxygen than the maternal hemoglobin when at lower partial pressures of oxygen. So when there is less oxygen available, when we're at a low oxygen tension, the hemoglobin of a fetus, the fetal hemoglobin, has to have a much stronger affinity for what oxygen is there than the maternal hemoglobin. So in the placenta, the fetus will absorb oxygen from fluid in the mother's blood, lowering the overall oxygen tension. And what that does is encourage the further release of oxygen from maternal hemoglobin. So just to explain, if the curve shifts to the left, what that means for any low partial pressure, so I'll do this in, I'll use green just to make this stand out again. Let's say we're looking at a partial pressure of, again, where I've got a partial pressure of 3, if we use this purple line, if I were to go up here, you can see that at that particular given partial pressure, the fetal saturation is much higher than the adult saturation. The hemoglobin is able to associate with oxygen much easier. So in a way, the fetus is going to absorb oxygen in the placenta. It's going to lower the partial pressure. And when the partial pressure is low, the mother, the maternal hemoglobin, you can, as we see here, has a very low saturation of hemoglobin. So the oxygen actually dissociates readily. It comes off, if you like, the hemoglobin, which is ideal for a fetus, because at that low partial pressure of oxygen, the percentage saturation of hemoglobin for a fetus is much higher, so it's able to pick it up. It's got a greater affinity for that oxygen. So it's almost like a deliberate step. It lowers the partial pressure of oxygen, because at a lower partial pressure of oxygen, the fetus finds it easier to saturate the hemoglobin, has a stronger affinity for that oxygen. Another particular question just related to this that I'd like to talk about is related to temperature. So on the same graph, you may very well get, if I just draw just a smaller version of that here, if we have the partial pressure of oxygen on this side here, and the percentage of saturation of hemoglobin with oxygen on the left, so percentage, if I just put percentage sat on the y-axis here. Now you may get given two particular curves, one like this, and one that looks a bit like this. And you may be told that the temperature is 37 degrees C here and 45 degrees C here. And the question may be, can you suggest how exercise affects body temperature and explain how the graph below, or I'm looking at this example below, but essentially the graph I've just drawn, how that graph supports the idea that an increase in body temperature actually aids delivery of oxygen to exercising muscle tissue. Now this was a question that I've asked my own students, and it proved slightly tricky, but if we consider it step by step, when you exercise, the body temperature raises. When you raise that body temperature, you can see that for a given, again we use green, for a given partial pressure of oxygen, let's just say here, the percentage of saturation of hemoglobin is far less. So at a low partial pressure of oxygen, in the respiring tissues, if the temperature is raised, we have a much lower saturation of hemoglobin. So what we're saying by that is that the oxygen is able to dissociate far more readily, and that's what we want. If we're exercising, we want our oxygen to dissociate readily from hemoglobin, especially when we're in respiring tissues, because those cells in those tissues need that oxygen for respiration. So when you exercise, there's an overall increase in the body temperature. The raise in temperature means that for any given partial pressure of oxygen, we've got a lower saturation of hemoglobin. So when there's a low partial pressure of oxygen in respiring tissues, for example, we've got an even lower saturation of hemoglobin. So hemoglobin finds it harder to pick up oxygen, it finds it easier to dissociate, so it readily gives up the oxygen which those cells can use. And that's how you should approach this particular question. So this one here just relates to the oxygen dissociation curve and body temperature. But again, it's just using a fundamental understanding based on this particular graph here, the general S-shaped oxygen dissociation or dissociation curve. Okay, hope all that helps.